Hello guys, good morning. In this lecture today, I'm going to talk to you about virus. This lecture is dedicated to the beginners who are studying in virology. Okay, let me first begin by the term virus. What does it mean? The term virus actually is a Latin neuter, which means poison. Okay, the term virus is a Latin neuter, so it's derived from the Latin word, so which means poison. Okay, this is what you need to understand. Okay, and then the study of virus is called virology. So branch of microbiology which deals with the study of virus is called virology. Okay, this is the two basic things you know. Okay, it is derived from the Latin word which means poison, and the study of virus is called virology. So now coming to the next point, what is a virus? Virus is an infectious obligate intracellular parasite comprising genetic material, DNA or RNA, often surrounded by a protein coat, sometimes a membrane. So I will break this definition for you. An infectious particle. Infectious means that virus can pass from cell to cell or from organism to organism. That's why this is an infectious. Okay? So obligate intracellular parasite, it means that the virus must infect the cell in order for its reproduction. Virus cannot reproduce on its own. For the reproduction, virus must infect the cell. That's why it is an obligate intracellular parasite. Parasite meaning that it takes the nutrients. Parasites are the organisms that take nutrients from other and harm them or damage them. Okay, so an infectious means it passes from organism to organism and cell to cell. Obligate intracellular parasite meaning that it has to infect the cell in order for its reproduction, and the parasite meaning that it takes nutrients from the cell, from other organism. Okay, and the genetic material. The genetic material of virus is either DNA or RNA. Okay, but the most of the viruses are RNA viruses. This one you have to remember, guys. Okay, the most of the viruses are RNA viruses, so their genome contain RNA, but some are also DNA viruses, so their genetic material is either DNA or RNA. And these particles are often surrounded by a protein coat. Like here I can say this is a protein coat. This is also called capsid. Most of these are surrounded by a protein coat. You have the genetic material DNA here, and this is the capsid or a protein coat. Okay, so most of the viral particles are surrounded by a protein coat, but sometimes some viral particles are surrounded by a membrane. For example, the example here. Okay, you see you have this genetic material RNA here, and it is further surrounded by the capsid and followed by the membrane here. Okay, so this is envelope or the membrane. Some viral particles are surrounded by the membrane, but some viral particles are naked, and these are called viroids, about which I'll talk to you later. Okay, so the definition of virus is an infectious obligate intracellular parasite comprising genetic material, DNA or RNA, often surrounded by a protein coat and sometimes a membrane, and sometimes some are really naked. They just have the DNA or RNA, just the genetic material, and those particles are called viroids, about which I'll discuss later. Okay, so I'm going to give you the examples of some viruses. So let's say the viruses which are enveloped or which have this membrane. They include dengue virus, influenza virus, and measles virus. So you see here, this is the membrane, the envelope, and these viruses, they have the membrane. Okay, and some viruses which do not have the membrane, they mostly belong to the Picornaviridae family. For example, hepatitis A virus, poliovirus, or Coxsackie viruses, they are non-enveloped viruses. Okay, so this is the introduction on viruses. Viruses are an infectious obligate intracellular parasite comprising genetic material, DNA or RNA, often surrounded by a protein coat here, and sometimes by a membrane like here, and sometimes they are naked. Okay, so now let's talk about what is a virion. Virion is a complete virus particle which can infect cells. Virion is a virus particle, it's a complete virus particle which has not infected the cell yet, so that is called virion. So what is an envelope? An envelope is a lipid and protein membrane. Just look at the picture here. This membrane, lipid and the protein membrane, this is called envelope. Okay, so that is a lipid and protein membrane. What is tegument? Tegument is amorphous layer between envelope and the capsid. Okay, we have here, this is the capsid and this is the envelope. The amorphous layer between envelope and the capsid is called tegument. And what is capsid? Capsid, it's a protein sheet, it's a symmetrical protein layer around the genome. Okay, we have here, for example, this is our genome, DNA, and here we have this capsid. So this is a capsid. So what is a capsid? It's a protein sheet around the genome. Okay, so that is a capsid. Now let me talk to you about viroid. Viroid, they only consist of genetic material, only DNA or RNA. For example, this picture here is an example of electron microscopic picture of potato, spindle, tuber, viroid. So they just contain the genetic material. Okay, they are called viroids. So these terms you should know. And the core is genome and associated proteins. For example, the core for this is this DNA for bacteriophage virus. Core for our human immunodeficiency virus, HIV, is this. The genetic material and associated proteins, that is called core. Okay, these are the terminologies that you should be familiar with. Now let's talk about a brief history of the viruses. The first viruses ever discovered was the tobacco mosaic virus. Okay, this virus is called tobacco mosaic virus. Like in the picture here, this infects this tobacco plant. It's called tobacco mosaic virus. And in this discovery, actually, two scientists played a major role. The first scientist, named Dmitry Ivanovsky, he reported in 1892 that extract from infected tobacco leaves were still infectious after filtration through a Chamberlain filter candle. Okay, so this extract from the infected tobacco leaves were still infectious even after the filtration through this special Chamberlain filter candle. Okay, Dmitry Ivanovsky reported it in 1892. And in the year 1898, Martinus, it's hard for me to pronounce, this scientist, Martinus, he extended Ivanovsky's results on tobacco mosaic virus, and he was the actually first to develop the modern idea of the virus, which he referred as the contagium vivum fluidum. Okay, contagium vivum fluidum, which means soluble living germ. Okay, contagium vivum fluidum. So the modern idea of the viruses was actually given by Martinus, scientist named Martinus, in the year 1898, and he referred this as contagium vivum fluidum. Okay, soluble living germ. So this is a brief history of the discovery of first virus. And the first human virus ever discovered was poliovirus, and this was discovered by Landsteiner and Popper in the year 1909. Okay, so first human virus discovered is the poliovirus. Now, I briefly discussed about what is a virus and about its discovery. Now, I want to mention that every single day we eat and breathe billions of virus particles. Okay, the virus particles, they are everywhere, and these virus particles, they infect all living things on this planet. Okay, they infect basically everything on this planet. And also our genetic material carries the viral genomes. Okay, that's you should know. So everything on this planet is infected. Every living thing on this planet is infected by viruses, and we eat and breathe billions of viruses every day. Okay, I just want you to know this. Okay, so like I said, we eat and breathe billions of viruses every day, so which means that the number of viruses on the earth is staggering. We have a staggering number of viruses. So, for example, the number of bacteriophage viruses in the water of the world, that is 10 to the power 30. This is a lot, guys. This is a lot, okay? The number of bacteriophage viruses in the water present in the earth is 10 to the power 30, which is a lot. Okay, so another example I want to give you is on this, that the number of viruses is staggering. If you go to the sea, coastal area, and you take out 1 liter of the sea water, and that 1 liter of the sea water will have more the number of that will be more than the number of people on the earth. Okay, so this is a very big number. So 1 liter of a coastal sea water contains the number of viruses which outnumbers the people on earth. Okay, so the number of viruses on earth is staggering. So now let's talk about how infected are we, because there are billions of virus particles we eat and breathe. So we all are infected with viruses. We all are infected with viruses, and there are some viruses. For example, herpes virus. If we are infected once, it is going to be there with us forever. Okay. So for example, if we are infected with some viruses, as an example, herpes virus. If we are infected one, it is going to live with us forever. So I want you to know this. So we all are infected with the viruses. Okay. So now let's talk about microbiome. So what is microbiome? For example, every different organ system in our body that has certain bacteria, and they have the beneficial effects in our body. Okay, so every part, for example, in the gut there are some bacteria, in our eyes and mouths, there is some sort of bacteria, dedicated in some specific parts of our body, and that has some beneficial effects. So like we have microbiome, similarly we have virome. Virome means that we also have virus particles in different organ systems. We have, let's say, in the skin we have some viruses, and in gut we have some viruses, in blood, and in the respiratory tract. So like microbiome, we also have virome. So virus particles in every different organ systems of our body. Okay, that is the concept of virome. So now I said that we all are infected with viruses, and with some viruses they live with us forever. So I want you to have a look at this picture here. This picture shows the human genome, schematic representation of human genome, which contains about 3.2 billion bases, about which only 1.5% they are protein coding. So, but I want you to know that there are some part of our genome actually contains viral genomes. For example, this part here, LTR retrotransposomes. So these 8.3%, these actually, a fraction of these contains the genomes from retroviruses. Okay, so by showing this picture here, what I want you to understand is that some part of our genome contains viral genomes. For example, this 8.3% of retrotransposomes, some fraction of it is actually from the retroviral infection, okay, from the retroviruses. So now I want you to talk about. I want you to know about that the viruses are one of the major reasons behind the global deaths. For example, if you look here, the respiratory diseases, in which some of those diseases were, of course, by the viruses that accounted for the death of 3.91 million people. And some diarrheal diseases, some, of course, some because of some virus, not only because of viruses, for sure, where the viruses also played role in these. 1.57 million people died, and because of HIV/AIDS, this virus, almost 950,000 people died in the year 2017, and from hepatitis virus, 126,000 people died from the hepatitis virus, not only C, sorry, hepatitis viruses. So not only this, actually, in the year 2020 now, because of these coronaviruses, the death of the people is already over 300,000, as of today. So showing this picture here, I want you to give you the information that the viruses are one of the major reasons behind the global deaths. Okay, so that's why it's really, really important that we study about them. Okay, so like I said, the vast majority of the viruses that infects us, they have little impact on our health or well-being, because I said that we eat and breathe billions of viruses. So what happens to them? For some viruses, actually they just pass through us. For example, in the cabbage, if in some vegetables, for example, cabbage, we eat these, we cook these cabbage and we eat these, and the viruses are not active, they are inactivated, and they just pass through our feces. And there was one experiment actually done in the Washington, D.C. What they did is that they took the cabbage from 5 different supermarkets in Washington, D.C. and they found out that each serving actually contained 10 to the power 6 viral particles, pathogenic for this cabbage looper. Okay, so by this part of the slide here, what I want to give you the message is that majority of the viruses that we eat, they don't infect us, or they have very little impact on our health. We eat them and we just pass them. They are just passed through us. Okay. So another example, another thing that I want to mention here is that most abundant, human fecal virus is paper mild motor virus, the number of which is around 10 to the power 6 per gram dry feces. And that means in 1 gram of the dry feces, this paper mild motor virus is going to be 10 to the power 6. So you see this number, right? How much? In just 1 gram. So the synopsis of this slide is that the majority of the viruses that they have very little or no impact on our health or well-being, we just eat them or drink them or inhale them, and then we just pass, and they are just passed through us. Okay? So not all viruses are harmful. There are some beneficial viruses. For example, norovirus. These noroviruses were shown to protect the gut of the mice when they were given antibiotics. The protected gut bacteria that were killed by the antibiotics made the mice susceptible to the gut infection. But in the absence of these bacteria, these noroviruses were able to protect their host. So not all viruses are harmful. Some viruses are beneficial effects. For example, noroviruses. Okay? So our immune system, even if we are infected, we all are infected with viruses, our immune system actually protects us against most of the viral infection. We have this immunity that protects us against most of the viral infections. That's why not all viruses, make us sick, or they have very little or no impact. Most of the viruses, they have very little or no impact on our well-being or health. So now, another question that I want to address here is, are viruses alive? This is a very big question. For this, I have an answer. A virus is an organism with 2 phases. It has a non-living phase, that is an infectious virus particle, which is called virion. Okay, this can infect the cells. So this is one phase. But this virion, when it infects the cells, it can then reproduce itself, okay, in this infected cell. So when this virion, the virus particle, is like this in non-living phase, when it's not infecting the cell, it is going to be like this there forever. It's not going to do anything. It's not going to affect us. It's not going to harm us. But when this, and it's not going to reproduce, but when this virion, this infects this cell, and in the infected cell, this virus particle can reproduce, and then it can infect us, and it can cause the damage. Okay, so the conclusion of this slide is that virus is an organism with 2 phases. One phase is non-living phase, which is called virion, and when it infects the cell, this cannot reproduce, but when it infects the cell, it can reproduce and it can cause harm. Okay? So another property of the viruses, they are passive agents. They do not think, they do not employ, explore, or exhibit. They don't think. They are just passive agents. They are just there. That this is what I want you to know. And let's talk about the sizes of the viruses. Before the concept was that most virus particles, they are smaller than bacteria, but now this has been changed because of the research. Current research, they have shown that the largest virus particle, for example, one of the largest virus particle is Pandora virus, and it can be up to 1.5 micron in length. Okay, and this can also be visualized under light microscope. Before mostly the viruses are 20 nanometer to 250 to 400 nanometer in diameter. And in the earliest days, the idea was the viruses were people were able to see, the scientists were able to see only under electron microscope. But now, the research is going on, and the scientists are discovering in the largest viruses. One of the example is Pandora virus, and the size of which can be 1.5 microns in length. This is the picture of the Pandora virus, which can be visualized in the light microscope. And scientists are still discovering, more and more giant viruses. And in the future, who knows? We will even have the viruses even bigger than these, and so on. Okay. Now, this is a very, very important slide that I want to discuss here. Okay, I want you to really understand this here. Let's first look at this picture here. This picture here shows that how bacteria divide. We have one bacteria, it divides by binary fission, it makes 2, and it divides further, makes 4 and 8, and so on. So bacteria actually they divide by binary fission. This is the time and this is the cell number. Whereas the virus particle, they actually replicate by assembly of preformed components. So how this happens? We have the viral particles, and these viral particles, this has to infect the cell, and when it infects the cells, then it makes the components necessary. It makes the necessary components and assemble these components inside the infected cell, and they make new viral particles. So the division of viral replication is by assembly of preformed components. Let's look at this picture here. This part shows the number of infectious particles, and this is the time. We have the virus particle, and we infect these viruses. The cells. The cells will be infected by these viral particles, and when the infection happens, and inside the cell, the necessary components will be made right inside this infected cell. Necessary components will be made. That's why we have this eclipse period in this viral replication process. Okay, why this is eclipse period? Because after the infection of the viral particles with the cell, it takes some time to make these components. These components. Once these components are made and they are assembled, and the virus particle divide and replicate. The virus particle replicate. Okay, the eclipse period in the virus is because first we have to have the viral particle, and it must infect the cells, and then it makes the necessary components inside the cells, utilizing the cells machinery, and then it assemble those components, and we have now new viral particle. We have now new virus. Okay. That's why we have this eclipse period. Unlike, it doesn't divide. The viruses don't divide like the bacteria. Bacteria divide by binary fissions, but they have a different system, like I mentioned here. Okay, so they replicate by assembly of preformed components. They infect the cells, and utilizing the cells machinery, make the necessary components, assemble them. Now we have the virus particles, and now this can infect another cell. And this process goes on. The eclipse period is the time for the making of necessary components and using the cell's machinery and assembling them to make the new virus particle. Okay, make the parts and assemble the final product. Okay. So now I discussed briefly about viruses. Now this is a question to you people, guys. So my question is, which of the following is true concerning the bacterial versus viral replication? Options are: viruses must assemble using preformed components. Bacteria do not replicate via binary fissions as viruses do. Bacteria must assemble using preformed components. Viruses do not have an eclipse period. Viruses replicate by binary fission. Please leave your answer in the comment section. If you liked my video, please subscribe our channel. Thank you very much for your attention.